Hello everybody, I'm going to talk to you today about where LAM cells might be coming from before they're in the lung, and about what type of cell they might be. And also, why is it important for our research that we know what type of cell LAM cells really are? We know that LAM's caused by cells with mutations in the TSC2 gene, and we know a lot about the protein product of TSC2, which is called tuberin. We know what tuberin's doing in the cell, and because of this, we know that rapamycin is a good treatment for LAM and TSC. But this doesn't explain all the features of the disease. It doesn't explain why only women get LAM, or why the lungs are damaged. And the reason for this is the behaviour of LAM cells depends on two things. One is the mutation that they're carrying in TSC2, and the other thing is the type of cell that they are. We call this the cell of origin of LAM, and we don't know what it is. In fact, if I made a list of all the things we know about the cell of origin of LAM, it would be a very short list. We know one thing, really. The cell of origin of LAM must come from somewhere outside of the lung. We know this because LAM patients who've had a lung transplant can have a recurrence of LAM in their donor lung. So why is it important that we know what the cell of origin of LAM is? I'll tell you something about the way we do our research, and we're looking for new therapies for LAM, therapies that will treat patients who don't respond to rapamycin, and also drugs that might cure LAM rather than just stop it from progressing. And to do this, we do two quite different types of experiment. The first type of experiment is an in vitro experiment. Here, we grow cells in flasks, we keep them warm, we grow them in special media, they go in a special incubator, with the aim of convincing them that they're still inside the lung. Then we treat these cells with drugs, and we see how the drugs change the behaviour of the cells. We use two different types of cells for these kinds of experiments. Primary cells come directly from diseased tissue, so these would come from LAM lung tissue from patients who've had a transplant or a biopsy. And model cell lines don't come from the lung, but do have TSC2 mutations. The other type of experiment that we do is an in vivo experiment. Here we take a mouse with LAM and we treat it with the drugs that we found are effective in our in vitro experiments. But now we have to think about how well the drug is tolerated, how it's delivered, and what the dosage might be. So the in vivo experiments take us one step closer to being able to trial these drugs in patients. But there are some difficulties with LAM research. No one has been able to isolate primary LAM cells from LAM lung tissue. We don't know why. Mice don't get LAM. We don't know why. But this leaves us very dependent on model cell lines for our experiments. And the model cell lines are not based on the true cell of origin of LAM because we don't know what that is. Now, if we do know what the cell of origin is, we can now make better cell models. We can now see if there are other ways to grow the cells. If we knew what the cell of origin was, we might know better what the culture conditions that it prefers are. And we might even be able to engineer a mouse that gets LAM. So you can see it's really important that we find out what the cell of origin of LAM is. I'm going to outline a few ideas as to what this cell might be that have been bouncing around for a while. The first idea is that these cells come from the neural crest. And this is based on the fact that neural crest cells produce PMEL and LAM cells produce PMEL. There's been a little bit of work on LAM as neural crest cells, but not lots. The second idea was that angiomyolipoma could be the site of origin for LAM, but not all LAM patients have an angiomyolipoma. Now, the favoured idea seems to be that LAM cells come from the uterus, and I'll explain why. Well, first of all, LAM is a female-specific disease, and only women have a uterus. Also, if you look in the uterus of women with LAM, you can find little lesions that look like LAM lesions. If you generate a mouse that has TSC2 null cells in its uterus, like LAM cells, then these cells can move to the lung. And there's a new paper coming out soon which uses some new techniques to address this question. This is the paper. It's not quite out yet, and it uses a technique called single-cell sequencing to look for the cell of origin for LAM. And I'll show you how they do that using Smarties. So this is the cells in your lung modelled using Smarties. There are lots of cells performing lots of different functions, expressing lots of different genes, but there aren't many different types of cell. If you could see the different types of cell, it might look a little bit like this. And amongst these cells are the LAM cells. What we can do now is take single cells from a lung and find out which genes that cell is expressing. And this is single cell sequencing. As you do this with more and more cells, you find cells that are similar to each other. And as you carry on, you can find a number of clusters of cells that represent the whole population of the lung. This is a real set of data from one of these experiments. The sequence data has been fed into a computer which has generated these clusters. Now, we know quite a lot about different types of cells in the lung and the genes they express. And using that information, we can identify which cells these clusters represent. But we're interested in one cluster in particular, and that's the LAM cell cluster. We know one gene that LAM cells express that no other cell in the lung expresses. And that gene is a gene that encodes PMEL. So the authors of this paper have identified, using single-cell sequencing of cells from LAM lung tissue, a cluster of cells that expresses PMEL. They've called these the LAM core cluster because these must be the LAM cells. And now we also know other genes that are expressed in LAM cells that we didn't know before because these genes are in the same cluster. And now we can ask another question. We can ask, are there any normal cells from outside of the lung which express the same kind of genes as LAM cells do? And when we do this analysis, the answer is that LAM cells are most similar to cells from the uterus. This is a really nice paper and it gives us lots of really new information about LAM. But there is one issue really with the conclusion that LAM cells must come from the uterus, and that is that men do get LAM. It's incredibly rare in the general population for men to get LAM, but in men with tuberous sclerosis it's actually not uncommon, but the disease never really progresses to the point at which it's symptomatic. So if men can also get LAM, but men don't have a uterus, then the uterus can't always be the cell of origin for LAM. So we have an alternative hypothesis, and our hypothesis is that LAM cells come from the pleural mesothelium. Now your lungs are covered in a thick layer called the visceral pleura. Then there's a layer of fluid, and then there's another thick layer called the parietal pleura. On each of those pleural surfaces there's a layer of cells, and these are called mesothelial cells. You might be able to see them in the top picture as a sort of dark purplish line. Sometimes when we get a piece of lung tissue from a LAM patient, it still has a bit of pleura attached to it. And sometimes on that pleura we can see mesothelial cells. As part of Suzanne's LAM immunohistochemistry project, I've been looking at the pleura in LAM patient samples. And sometimes in the pleura we see regions that look just like LAM lesions, just like the LAM lesions we see in the lung, but they're embedded in the pleura. They express all the same markers, even PNL2, which is specific for LAM cells, and I've circled one of these lesions in this figure. Sometimes in one of these samples there are some mesothelial cells, and if we look in the mesothelial cells we can see LAM markers there too. Now, it doesn't make biological sense that these cells on the outside of the lung could have come from LAM lesions inside the lung. What makes more sense is that the mesothelial cells on the outside of the lung have travelled into the lung to form the LAM lesions. In fact, that mesothelial cells are the earliest stage of LAM in the lung. So this is our model for how LAM might develop. We think the TSC2 mutations happen on pleural mesothelial cells on the outside of the lung. Because these cells have lost tuberin, this changes their properties, and now they divide much more quickly and they become sort of invasive. We think they can invade all the way through the visceral pleura, and then they can migrate into the lung. And because they have dysregulation of mTOR, because they've lost tuberin, we think they start to destroy the lung because they produce too much protease activity. This is a nice model because the properties of LAM cells are similar to the properties of mesothelial cells. Mesothelial cells are outside the lung. They can, even in normal development, enter the lung. They produce VEGF-D, and they can even, quite surprisingly, produce fat cells, which means that mesothelial cells could produce angiomyolipoma. And what we need to do now is test our ideas experimentally. As Suzanne mentioned, we now have a new home. We are based in the Biodiscovery Institute on the University of Nottingham campus, which is a brand new building. It has fantastic facilities for us to do our work, and it's also brought us in contact with a lot of new research groups. LAM is an interesting respiratory disease because to understand it properly, you need to know something about cancer biology and something about stem cell biology. And in the Biodiscovery Institute now, there are many cancer and stem cell biologists that are our new neighbours, and we can use their expertise to address the questions that we have about LAM. And our first collaboration is with Dr. Nick Hannon, who's in the Division of Cancer and Stem Cells. And he's going to work with us to use a technique called CRISPR-Cas9 to delete TSC2 from mesothelial cells. And what we want to know is, if we do this, do mesothelial cells turn into something that's very much like a LAM cell? So by this time next year, we might have been able to build a new model for our LAM studies.
Thank you, Debbie, for a fascinating presentation. And yes, in terms of questions, you should be able to hear me again, hopefully. Yep, all good. I just wanted to ask, was it just by chance that when you were looking at pleura cells, you discovered how similar they are to LAM cells?
I was interested in the pleura for two reasons, really. One is that LAM patients have a very high rate of pneumothorax. So that made me think that maybe there was some underlying issue with the pleura that made them more susceptible to pneumothorax. And the other reason, it came from some work that we were doing on angiomyolipoma. We're interested in how a cell, a LAM cell, can have two completely different phenotypes in two different places. So it forms cysts in the lung and it forms fat in the kidney, the AML. So I was looking at where the origins of renal fat might be, and it turned out to be the mesothelium. Not the pleural mesothelium, but the peritoneal mesothelium, but essentially the same cell type. So then I started to look more closely about what pleural mesothelial cells were doing. And we found the LAM markers in the pleura and in the mesothelial cells, which is actually a very significant observation, because it means these must be LAM cells.
Yeah, okay. And so talking about mesothelial cells, they seem to be like kind of wriggly little things that can kind of get through from the surface and from the pleura down below. And actually, I too had that same thought. Have they then left a kind of trail of destruction wherever they've gone through into the lung, which is why you can get leaks into your lung cavity, as it were?
Well, that's a good question, and I think that might be true. So mesothelial cells, their normal role is just to make the fluid that makes the two pleural layers slip across each other. But during development, they do all sorts of different things, including moving into the lung to form mesenchymal cells, which is what LAM cells are, and forming fat around the kidney. So they already have the properties that you need from LAM cells. They just need to be sort of reactivated. And what we think is that loss of TSE2 and loss of tuberin means that mTOR goes a bit wild, and it reactivates these pathways that mesothelial cells would do under normal development, but now they're doing them in a pathological, in an uncontrolled way. And so, yeah, I think as they go through the visceral pleura, they damage the visceral pleura and they leave it vulnerable to pneumothorax.